Welcome again, guys. We're talking about biology problems, and here is another problem from the polymerase chain reaction or PCR reaction. Very important problems. You need to understand this for practical purpose and also for other purposes. So let's see the question. That is a 200 microliter of PCR mix has 100 template DNA molecules, and the reaction was performed for 10 cycles. So now they asked us two questions. One is how many molecules of amplicons will be generated. Second is how many molecules of amplicons will be present in 0.1 microliter of the reaction mixture. Here this is that doesn't mean mole, it is molecule. Okay. So in this case, in all this type of PCR problems, the easy thing to understand is how PCR works. That is, for PCR, if you have one DNA molecule, it will make it 2. If you have 2, from 2 it will make 4. So it is an exponential part. So from 1 it becomes 2. So let me write here in this way. From 1 it becomes 2. From each of those single one it becomes more, so 4. So from this 4 you can just see the exponential growth becomes 8, like so on. It will produce more and more DNA. So the number of increment is having a pattern, and that pattern can be fed into a simple equation, not equation, a simple formula. And the formula for calculating the number of molecules of DNA that is generated after a particular number of cycle, after say n number of cycle. So let's assume the number is n, the number of cycle. The formula states 2 to the power n, because 2 is the number, this is the times, so twice number of DNA molecules are present from particular one template. So here is the power that is going to be multiplied each time. So if you have one DNA, it becomes from 1 to it becomes 2 twice again. From 2 it becomes 4. So from 4 it becomes 8. So it is increasing in the multiplied by 2. So twice increment in all these different stages, all these different cycles. So it progresses each cycle and it produces twice of that DNA molecule that is present in the previous cycle. So that's why I put 2 here, and n is the number of cycle that is being carried out. Now in this case, what is the number? That is 10 cycles that is being carried out. So he puts 2 to the power 10. So that is going to be the number of template. But remember, in all this case, if we assume at the starting point we have one DNA template, right? Because if you have one DNA template, that is obviously beginning, so 1 multiplied by this, because this template always present. So now this value is being multiplied by each of these numbers. If the DNA template for the start is 1, we put 1 here multiplied by 2 to the power n. So 1 into 2 to the power 10, and that is going to be the number. But remember here, they suggest us that PCR mix contains 100 template DNA already. Right? So actually it never consists of one DNA, right? Because we add some concentration of DNA there as a template. So here the number of DNA as taken as the template DNA at the starting is 100, not 1. So what we'll put now, that is going to be 2 to the power 10, but instead of 1 we're going to multiply it by 100, because that is the starting concentration, isn't it? So as that is the starting concentration, what we know here is that 2 to the power 10 means 1024 multiplied by again 100, so 102400. That is going to be the number of DNA templates generated after 10 cycles of the PCR, starting with 10 DNA templates. 100 DNA templates. Why we are multiplying 100, not adding 100? The reason is that from each of those 100 DNA molecules, each of those 100 DNA templates, it produces this exponential growth. So why should we add that 100? Because each time from each of these 100 DNA molecule, we are getting twice number of DNA. That's why I need to multiply this with 100, not adding 100. That's a very important thing to understand. So once we know this, the second thing is how many molecule of amplicons will be generated and will be present in 0.1 microliter of the reaction mix. Now for that, they have provided the idea of a 200 microliter reaction mix. Now once we have 200 microliter reaction mix in the position, so whatever number of DNA that is generated, that is here 102400, that is the number of DNA molecules that is being produced after 10 cycles, starting with 100 DNA templates. So here 200 microliter DNA contains this number of DNA, right? 200 microliter PCR mix contains this number of DNA, right? 200 microliter contains this number of DNA. Now we need to know the concentration. The concentration of DNA in 200 is this, right? So now what is going to be the number of DNA molecule in 0.1 microliter? Very easy. So for 1 microliter of the DNA, it is going to be 0.2400 by 200, and for 0.1 we again multiplied by 10 here. So ultimately what we'll get here, we get the value 51.2. I've calculated it earlier, so I know that this 51.2, and this calculation is very easy. So if we have this number of DNA molecule in 200 microliter of PCR mix, so what is going to be the concentration in 0.1 microliter? They are all put in, in a microliter, same unit, so no unit conversion, no problem, no complications, fairly easy calculation. That is going to be the number of DNA molecule that is present in 0.1 microliter of the reaction mix, because that is also very important to know, because sometimes during this experimentation and after PCR we need to take some concentration, so some amount of those PCR mix and run it in some other way or use it for other purposes. So we need to know the concentration of DNA that is present in that PCR mix. To understand that, we need to calculate these things, right? So this in a sense is one of the very important problems that you should know about if you're doing in molecular biology lab or any cell biology kind of lab, because nowadays PCR, it is the ultimate reaction machine that should be present and it should be required in most of the field of biological processes. So you need to know, calculate these things.